this week we're going to be talking about mutations. Specifically, the objectives for today's lesson are what are mutations, what are examples, and their effects. And lastly, we're going to talk about what are some diseases and applications of mutations and uses in the lab. So by the end of today, you should be able to take care of mutations, know what they are, a definition of them, some examples of them in terms of how they affect your DNA, and their effects on the phenotype. What are some examples of diseases that are caused by mutations, and applications and uses in the lab? And so that's what we'll cover today. All right, so we'll start with our first question. What are mutations? Mutations simply are changes in the DNA. So a lot of times when people think of mutations, they think of things like Teenage Mutant Ninja Turtles, X-Men, all kinds of things. But mutations are just changes in the DNA. They can be good, bad, or neutral, meaning they have no effect on the individual. So they can be good, bad, or neutral. In terms of types of mutations, you can think of mutations that can be very large. And so these are ones that happen on the chromosomal level. So remember, we have a number of chromosomes. So if you have an extra chromosome, and an extra chromosome would happen as a result of a problem or a change in meiosis. It could be a deleted chromosome or a missing chromosome. An example of this would be trisomy 21 or Down syndrome. They can also be considered large because they would be a change at the chromosome where you're completely missing it, or in recombination you would have an error. So an example of this might be here is your chromosome. You have one black chromosome and one blue chromosome. And those would be the direction. They'd have a directionality to it, where it has a top and a bottom. Maybe in recombination, after, you would end up something where the chromosome would flip. So now instead of it being oriented down, you would have an up-facing chromosome. Like this. And so we note the change here because it flipped. This would be an example of a translocation or a really large change. These are examples of large changes. So the vocabulary word for that would be a translocation. So we have large deletions, large additions, and translocations where they physically move. The opposite of large would be what? Small. So we have small mutations as well, and small in terms of where its effect is. They can still have huge effects in terms of phenotype, but small as in that it's not a huge chromosome change, but rather a DNA change. And so these things can have large effects, but we have something called a point mutation. And the reason why it's called a point mutation is because it happens at one point. It occurs at a single nucleotide. So an example of that, what are the four nucleotides? Remember A, T, C's, and G's, and just those nucleotides change. A's, T's, C's, and G's. So for example, if we're looking at the DNA, you had A, T, C, G as your sequence. A single nucleotide changed maybe something like A, T, T, G, where this C is now a T. This would be a point mutation that's changed. In addition to point mutations, you can have deletions and additions, again, changing at one point, but they can have effects called something like a frame shift. And a frame shift has to do with the reading frame. If you recall back to gene expression, we're reading groups of 3 called codons. And because of that, if we have a deletion or an addition of a non-multiple of 3, so for example, A, T, C, G, A, T, so we have A, T, C, G, A, T for our sequence. We'll go a couple more nucleotides. If we were to delete just the G, so we want to delete just the G, our new sequence would now be ATC would remain the same, but now it would be ATTCA. We've caused a change right here, a frame shift, where we've read in groups of 3, the entire frame of the DNA has shifted down. And that's an example of a frame shift. We also break down mutations into things that are sense mutations and non-sense mutations. A sense mutation is a change of amino acid to amino acid. So this would be really common in things like a substitution, a point, and even a frame shift. You're changing amino acids. This is in contrast to a non-sense mutation. Non is a prefix meaning no, so it means a no-sense mutation. It doesn't make sense. We're going from an amino acid to a stop codon. So a stop codon causes the DNA to stop being transcribed or translated. So instead of it being an amino acid, we now have a stop codon, and this creates issues because it creates things like shorter proteins that are non-functional. So in terms of which mutation would be the most dangerous, we would have things like frame shifts followed by nonsense, because now you don't have a protein at all. That protein is now missing, which can create major problems. As I said earlier, there are 3 kind of things that can happen as a result of a mutation. It can be a good thing. In terms of evolution, it can lead to a trait that makes it better able for you to survive in the environment. It can be a bad effect, meaning it's detrimental and it causes a disease state. Or third, it could be neutral and have no effect. I'm going to focus on this idea that a mutation can lead to a disease. And so how does that happen? A lot of times it leads to a disease because it's a non-functional protein. So we talked about that with a nonsense mutation. But the idea is diseases can be a result of a protein that's non-functional. For example, if you normally needed an enzyme, think about an enzyme. Remember, they can build or break. If you have an enzyme for consuming sugar, if you don't have this enzyme for consuming sugar, you are now no longer able to consume sugar. We talked a little bit about albinism. Albinism could be another example where you no longer have pigment. It could lead to a non-functional or decreased function of an enzyme. For example, lactose intolerance is a result of a non-functional enzyme for lactase. So a lot of these diseases are the result of you not having a functional protein, meaning you're unable to do what you need to do. In terms of benefits, positives of these, things that can lead to faster metabolism, a better protein, and we also talked about things that were neutral. Neutral effects would be because it's the same protein or a silent mutation. Meaning the mutation is present, you have a change, but it causes no change in the final protein because the amino acid does not change, or because the folding or the functionality of this protein, so we start out with the protein like this, and it folds into something like that, remains the same. So a structure here may stay here, and it makes no difference. So it's really important to understand that mutations can be bad, good, or have no effect. So that was kind of a quick run through some diseases. We'll learn a lot more about them in class in terms of why they're inherited, how they're inherited, what are their effects on individuals. Lastly, I'll touch on, just briefly, the applications of this use in the lab. Lastly, we'll touch on mutations in the lab and why they're useful. So in a lot of ways, people think of mutations as a bad thing. If you go outside, you can get cancer, you can have problems from the sun, UV rays. But in terms of a lab, you might actually want to generate mutations. Because you can generate mutations, so one way is with mutagens, something that causes mutations, the one being UV rays. You can generate mutants in a lot of different types using these UV rays, and this is to create either diversity, you can create genetic libraries, and you can kind of force evolution. So creating mutations is a great way to test for evolution. And it's a great way to test traits or test treatments in terms of medication. So one example you might have seen this in if you were watching what is it? I Am Legend, he creates a bunch of these treatments so that you can then test it. We talked about the obese mice, where you have knockout genes. So you can create mutants that are called knockouts, meaning they are missing it on purpose. And this can help you learn more about science because you can find out the uses. So learn protein pathways. And you can kind of figure out what's going on. If I knock this gene out, what does it do? And this is really common in trying to learn more about the genome and kind of more about science. And you can do that in a great way by using mutations, specifically knockouts, because you can learn what happens when something is missing or something is added. So you can either add, delete, and modify. And this is when we're using a lot of our test organisms, things like mice, things like C. elegans, things like nematodes, a lot of worms that we could use these mutations in to kind of see what their effects would be. So in summary, these are kind of the 3 big points. We know that mutations are changes in the DNA. They can be big mutations, such as a chromosomal shift. They can be small mutations, such as a point mutation or a simple deletion. They have the benefits in that they can help you evolve, for example, letting you blend in more with your environment, making you be faster, have more of a certain muscle type. They can create problems, such as disease states like cystic fibrosis, where you're not able to breathe as well, causing things like lactase problems where you're not able to drink milk as well. And they can have no effects, and this would be an example of a silent mutation where there's no effect on the final protein. Diseases are generally caused by a loss of function, meaning that something that you were able to do you're no longer able to do. And lastly, we learned about how mutations can be used in the lab. They're generally used to find out more about science by creating a knockout. If you knock something out, you can then find out what it does later. So hopefully you learned some stuff about mutations. If you have any questions or you're still lost on what's going on in mutations, whether it's a question about its use in transcription or translation, or how it's used in science, please feel free to shoot me an email, give me a call. I don't bite. So hopefully this was helpful. If not, always, of course, ask questions. Hope you have a great one. I'll talk to you guys later. Have a nice evening.